Hi, everyone. Happy Friday. Welcome to the Myotonic Dystrophy Foundation's Meet the Drug Developer webinar. I'm Tanya Stevenson, the CEO at the Foundation. We are delighted you are joining us today to hear from AMO Pharma. Just a bit about the Foundation. We envision a world with treatments and a cure for myotonic dystrophy. Our mission is community care and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care. We accelerate research towards treatments and a cure. A reminder of some of the resources and support that are available on our website. Our toolkits and publications, including clinical care recommendations, are found at myotonic.org/toolkits-publications. Our support programs, including our support groups, information can be found about those at myotonic.org/find-support. Our calendar of events is at myotonic.org/calendar/month. And our digital academy, which hosts our presentations and webinars from the past, including past conferences, can be found at myotonic.org/digital-academy. Today's presentation is being recorded, and we will post it to the Digital Academy within the next week. Just a reminder that we have been hosting these primarily on the first Friday of each month at 12 o'clock Pacific. And while today we are hearing from AMO, next month on August 4th, if you are not already registered, we will be hearing directly from Avidity Biosciences. We will also hear from 10 different companies working on treatments and a cure for myotonic dystrophy at our conference in Washington, D.C., September 8th and 9th, and we hope you will join us there. You can find out more about the conference at myotonic.org/conference. Okay, today we are going to be hearing from two folks from AMO Pharma. Dr. Joe Horgan is a pediatric neuropsychiatrist and has specialized in the treatment of children with complex neurodevelopmental disorders for more than 30 years. He was a senior director in the Neurosciences Medicines Development Center at GlaxoSmithKline, where he also co-funded and led the company-wide Medicines for Children Advisory Network that collaborated with all therapeutic areas in the company. Dr. Horgan also served as Assistant Vice President and Head of Medical Research for Autism Speaks, the largest science and advocacy organization in the U.S. devoted to autism spectrum disorders. He subsequently served as Vice President of Clinical Development and Medical Affairs for Neuron Pharmaceuticals Limited, leading the company's pioneering clinical development efforts in neurodevelopmental disorders such as Rett syndrome and Fragile X syndrome, as well as in traumatic brain injury. Also with us today from AMO is Dr. Mike Snape. Dr. Snape has more than 30 years of experience in pharmaceutical research and development and has managed relevant projects from preclinical through clinical stages of development. He previously worked in CNS research and development at AstraZeneca and also has extensive experience of research and development projects in the field of CNS disorders in a small life science company environment, having been principal scientist at Cerebras Limited and associate director of Vernalis, and was a founder of Neuropharm. Dr. Snape has been an advisor to venture capital groups in the UK, the Wellcome Trust, and sits on the advisory council of multiple research foundations. He initiated one of the first industry-sponsored clinical projects in autism in 1997, led the first industry-sponsored multi-center studies of a core symptom of autism, and some of the first industry-sponsored studies in Fragile X syndrome. More recently, Mike was one of the founders of AMO Pharma. Thank you all for submitting your questions in advance. The questions have been provided to our speakers today. They have done their best to integrate them into their presentation. If you have questions during the presentation, you can type those in and send them to our speakers. They will be answering some questions at the end of the presentation. To do that during the presentation, just go to the Questions tab, type your question in, and click Send. If you are on an iPhone, just click that question mark icon at the top of the screen, type your question in, and click Send. MDF works very hard to make sure that we can bring relevant information to our community. If this program speaks to you, please consider a donation, because together we are changing the future of myotonic dystrophy. Okay, without further ado, it is my pleasure and privilege to welcome the AMO Pharma team.
Good morning, good afternoon, good evening. Thank you, Tanya and Clede and everyone at MDF for the opportunity to speak today. And thank you to everyone from the community who's listening to us. I'm hoping that my screen is showing at the moment. Joe, can you confirm it is?
Yep, we can see it.
Cool. If you give me 1 second, we'll move to it. All right. Thank you so much again, everyone, for being here today. We are going to present this evening a little bit of information about a trial that we have just been running on a molecule, potential novel medicine we call AMO02. We have just finished up a study in congenital myotonic dystrophy. I'll give a slight apology at the beginning. We were targeting our ambition to actually present data on whether or not the drug had produced some benefit at this meeting today. Unfortunately, we are delayed in getting all of the data into the analysis process. So we'll give you some little snippets of information, but we'll have to come back another time to give you the full data. So Joe and myself will go through what we have. I'll start off, pass over to Joe in a moment, and then come back to me at the end. And then we're happy to take any or all of your questions when we get to the end of the presentation. So we have been focusing on congenital myotonic dystrophy, children with DM1. The real reason why we started off there, I think the clue there was in Tanya's introduction. Joe and myself, for personal reasons as well as professional reasons, are interested in childhood onset developmental disorders, and our principal experience is in assessing developmental disorders in children, and there are clearly developmental issues for individuals who have the congenital onset form of DM1. We hope we've been gaining experience in the neuromuscular disorders and myotonic dystrophy generally, and that will chain forward into the design of future studies we'll mention them in a moment or two. But for today, we're talking about congenital DM1. I know there will be individuals with DM2 in the audience, and we've mentioned before, we know that you're there. We are trying to explore whether AMO02 could be helpful in DM2, but we don't have answers on that just at the moment. And equally, I think there's already questions around, will we be thinking about adult onset DM1? And yes, we are, and we'll mention that in a moment. As it happens, when we're talking about congenital onset, the things that we can measure in clinical studies, like the ones we're going to describe now, are the symptoms that are present all of the time in the condition. Those cognitive deficits, developmental delays, motor issues, and I use the term motor deliberately, muscular issues, and bodily issues. It's obviously more difficult to try and capture the more episodic, sporadic things, but we understand that those other complications of congenital DM1 in its very difficult and varied presentation are there. It's just difficult to always capture all of them in a clinical study. Obviously, there are no treatments for DM1, and I don't need to tell this audience that the causative agent in the myotonic dystrophies is the presence of a pathogenic or abnormal RNA. So what is AMO02? I know we've discussed this before, but for new people in the audience, it might be worth going over it. AMO02 is an experimental medicine. It's not approved for use in the treatment of any patient group as yet. It presents as a little sachet of powder that you pour into water, and it makes a strawberry flavored liquid, and then you drink it, or you administer it through a feeding tube if that's what you have to do. Once you take it orally, it goes through the digestive tract, enters the bloodstream, circulates around the body, goes into all of the organs of the body, so far as we know, including the brain and muscle, and then enters cells. And when it enters the cells of the body, you see what's shown in these pictures here. These black squares in the middle of the screen show either muscle fibers taken from a young child with congenital myotonic dystrophy, or the equivalent tissue taken from one of the mouse models of the condition. And you can see this fluorescent red staining is a method of visualizing the DMPK RNA that causes myotonic dystrophy. And as you can see, in the presence of AMO02, it goes away. And that's functionally what AMO02 does. It attacks and reduces the causative agent of DM1. And it does that, theoretically, at least in all tissues of the body. And if you want to read about that, there are some papers that have been published in the literature where you can get a handle. We had previously done an initial small exploratory study, a phase 2 study, if you like, in adolescents and adults with childhood onset DM1. It was conducted in the UK. And the short version of that is we measured lots of different things using lots of different measures, and we'll come back to outcome measures later in this talk. But the short version is whatever type of measurement system we used and whatever organ system of the body we targeted, the drug seemed to offer some encouragement and improvement. So that was the stimulus for us to move on and do a second study, a larger, more broad-based study. And we're going to discuss that next. And I'm going to pass over to Joe to talk you through the next 3 slides on the detail of that. So Joe, if you're still there, over to you.
Yep, no, I'm definitely still here. So this is the study that we just finished. There's 2 different names that we've used. Our internal name is the 003 study, which is quite boring. The external name that we've used is the REACH CDM study, and the CDM, of course, stands for congenital myotonic dystrophy, and REACH is our acronym for the drug and the design and what have you. This is a study that was larger than the initial study that was done in Newcastle in the UK that Mike just talked about, purposefully because it is one that has enough patients or subjects in it. We use the term subjects for these types of clinical trial pieces of research, but it had enough subjects in it to make it appropriate to interact with regulatory agencies like the Food and Drug Administration and their peers in the United Kingdom and Australia and Canada to discuss the design and the outcome measures and then the results. So that's the reason that we use the term pivotal. We've just finished this study, as Mike had mentioned, and the results are still being analyzed, but I'll give you a snapshot of a few things that we already know in terms of what transpired. The study took place at sites that were located in the US, Canada, Australia, and New Zealand, so 4 different countries. And it involved both children and adolescents with early onset myotonic dystrophy, well, in particular, congenital onset myotonic dystrophy. So symptoms were present at birth. CGIS greater than equal to 4 is just a method. It's a particular rating scale of severity. It's a method that we used to assure that the youngsters that were included in this study had sufficient signs and symptoms of their congenital myotonic dystrophy such that we could show a change in the event that the test drug was beneficial. And it was a double-blind, placebo-controlled randomized study, so neither the investigators or the physicians, for example, nor the families and the children knew if they were being randomized to either AMO 02 or placebo. And it was a 50-50 randomization scheme with a 20-week active treatment period. We also have composed a companion study to this called the REACH-CDM-X study, also known as an extension study. The internal name for that is the 004 study. And it's a 52-week open label treatment period in which everyone that participated in the preceding REACH-CDM study qualified for being in the open label extension study if they had finished the 003 study. And we actually extended this REACH-CDM-X study recently, so it went an additional 32 weeks and now has an open-ended component for it at most of the sites that are involved in this study. The objective in that REACH-CDM-X study is slightly different in that we're looking at longer-term safety as well as doing some efficacy monitoring as well, looking at evidence of sustained clinical benefit. Can you go to the next slide, Mike? And this is just a snapshot of the size of the study who came in. I want to first thank every family and every child and adolescent, as well as the sites that helped us to put together this program and that opted to enroll in this program. We ended up enrolling 56 individuals at the very start of the study. 53 of them ended up contributing data to the results that I'll be showing you here in just a second. So not a large study, but large enough, I think, to be informative in terms of understanding what role this drug has with regard to young people with congenital myotonic dystrophy. The mean age was 11 years of age, so kind of evenly split between that age range of 6 to 16 years of age. More males than females. And then the rest of this, I think, we were somewhat surprised that we had more left-handed individuals than right-handed individuals. I'm not sure what to make of that. And then the number of CTG repeats, which is the key genetic issue that contributes to congenital myotonic dystrophy. There was the mean value was over 1,000, although the range was actually somewhat broad, from 200 to 2,200. And then the BMI, which is the value at the very bottom, basically just says that these children and adolescents were essentially normal in terms of their height and weight and what have you. Can you go to the next slide, Mike? And then this is just a snapshot of some of the safety and tolerability data, which is really the first cut of data that we've had available to us. We knew that the day-to-day experience of taking the drug was actually pretty good in terms of what it's like to take medicines, whether they're experimental medicines or approved medicines. There are several medicines out there, for example, that typically would cause things like nausea or headache or make you feel tired or maybe interrupt with your sleep. And those would be the types of side effects that would cause children and adolescents to want to discontinue from being in a study and what have you. And we were lucky in that we didn't encounter any of that. So the day-to-day tolerability of the medicine was actually really excellent. And that's played out here in these numbers where we show you the incidence of particular side effects on AMO 02 versus placebo. I put a bullet point in here where I'm using a filter of a side effect or an adverse event that has a rate that's higher than 10% and more than 2.5 times the rate of placebo, because that's a classic way that the FDA, for example, would decide on which potential side effects they would put in the highlights of prescribing information for a new drug. So for us, that was cough, headache, and rhinorrhea. And if you actually look at those numbers that give you the incidence of the side effects, they're quite low. So for cough, it was 14%. For headache, it was 11%. Nasopharyngitis just means stuffiness. And usually it's associated with things like cough and sneezing. And then moving down there, kind of to the middle of the list, rhinorrhea means runny nose. So, somewhere in that range of 10 to 15% for that cluster. But then when you look over in the other column at, for example, upper respiratory tract infections or nasopharyngitis on placebo, or at the very bottom, if you look at nasal congestion on placebo, more or less the same sorts of symptoms of upper respiratory tract infections. So it actually turns out to be somewhat even. One thing that is interesting here is that the rate of diarrhea it was 20% on placebo, but only 7% on AMO02, which in my mind meant that there could be a possible protective effect here from AMO02 in terms of the natural rate of GI problems that you'd have with congenital myotonic dystrophy. That's something that would need to be explored further. And then the only other thing I'll point out here is the 2 instances of laboratory values where there is an increase in what we call the transaminases or ALT. Those are lab values that tell us how the liver is functioning. And those were elevated in these 2 instances, and that was deemed to be an adverse event. They reversed themselves when the medication was ultimately discontinued, or just with time, they turned out to be to normalize on their own. But otherwise, the drug very well tolerated and no real significant highlights in terms of safety information. Do you want to go to the next slide, Mike?
Thanks, Joe. Thanks for that.
Do you want to talk for this? Okay.
Yeah, cool. Okay, so again, in an ideal world, we might have had data today. Because we haven't had all of the data inputted yet, then the independent person who does the analyses for us hasn't completed that. And I'm not going to take any guesses as to what's happening. That would be inappropriate. So we will update as soon as we can as to where we get with the analysis. But I thought it might be helpful to set the scene today to talk about the measures in the study. So you've got an idea of the kind of things we will be looking at and will be reporting on just as soon as we can. And I think it's deliberate and important that we didn't just pick a single measure. This is not like trying to develop a new medicine to treat high blood pressure, where effectively you just measure high blood pressure. We all know that congenital myotonic dystrophy, I hope we all know, is a complex condition. It can affect multiple organ systems, not constantly all of the time necessarily, depending on the system, with varying degrees of severity. It's very complex, very, very challenging, of course, for individuals. And trying to pick one thing to assess that is really, really tricky, and in our view, not necessarily appropriate. So, we've used this graphic before to try and think about many different organ systems and many different symptom domains that we need to canvass so that we can see what's going on with the entire presentation of a participant with that. So we're trying to measure multiple different things across the body and use multiple different people to assess them. And the kind of broad buckets, the 4 silos of information, are rating instruments. So those are systems where an individual will look and observe the individual being participating in the study and give them a score according to the severity of the presentation of their symptoms. And we've used a variety of different measures that try to canvass or assess the symptoms all the way across the body, completed by either the clinician, the investigator at the site, or the caregiver for the person who's participating in the study. So that's kind of bucket number 1. Then bucket number 2 is at the sites in the study, usually completed by a physiotherapist, looking at functional assessment of motor, i.e. movement, and muscle function. And I use the term motor because it's difficult for me in my mind to distinguish the control of walking and the ability to walk. It could be brain, it could be muscle, it could be the 2 mixed together. But we've looked at a standard test that I'm sure this community has heard described before, the 10-meter walk-run. And then we've looked at hand grip myometry to look at the strength of grip using a computer-based system to assess that. So that's one bucket of functional assessments. Then we've used functional assessments to look at cognitive skills and real-world function, again, commonly at the investigator sites in this study, performed by a neuropsychologist. So we've used something called PPVT, or Peabody Picture Vocabulary Test, which is a very commonly used test in children with developmental conditions used to assess their IQ and cognitive skills. And then we've used something called the Vineland Adaptive Behavior Scale. The Vineland is a very highly normed scale. It's used very, very extensively by child psychologists, pediatricians, to assess the level of real-world function as a child. And there are data points for thousands and thousands of healthy children and children with different developmental conditions, so that we can relate the performance of an individual to a broader understanding of children in general. And I just put in brackets one of the scales within that, the daily living skills scale. I mentioned that because previously we've run a study with AMO02 in a double-blind trial, very, very like this one, in children with idiopathic autism, where AMO02 improves performance compared to placebo. So it's a good comparator to have from that. And then the last clutch of measures that we've used are, well, I guess, what we call objective biomarkers. So things that don't necessarily rely on human intervention, like a specific kind of X-ray called a DEXA scan that can measure the amount of muscle and bone density in an individual, looking at various proteins that circulate in the blood that reflect the status and the health of different tissues in the body. And then also the amount of drug in the blood. Any drug taken orally doesn't necessarily achieve the same level in every individual. And one would expect the more drug you have in your system, the more marked or the greater the extent of any benefit that might be observed. So it's important for us to know that. As I said at the beginning, we have just about got all of that data in now, database. Some preliminary analyses are underway, and the full analyses we were targeting for the end of this month, but because of a little delay on some of these, we're spilling into next month. But I'll come back on timings at the moment in a moment. So those are the things that we've measured, quite a comprehensive picture. And I'll echo Joe's comments. It's a long day for the investigators, all of the staff at the site, the families who've had to travel, and of course the children undertaking the study. The amount of effort that people went to complete this study was truly extraordinary, not least because, of course, the impact of COVID. So I just want to extend my thanks as well to everyone who took part and participated, and all the patient advocates that helped us. So in conclusion, where are we? We're very near to completing the analyses. As soon as that happens, we will disseminate the data. We won't wait or delay. I'm sure MDF can help us put that message out. So we'll crave your indulgence for another month or so to get that data. We'll disseminate that as soon as we can. In the meantime, we mentioned earlier on, of course, this is our starting point. We are also planning on moving into adult onset DM1. We have designed a study. It's now out for commentary from the patient advocacy community and also from independent experts to look at the feasibility of that study. So we will present that just as soon as we can. And then with that study in mind and the data from the present study, the one we've just described, we will go to the regulators in different countries, starting with the MHRA in the UK and the FDA in the US, to discuss their understanding of the data that we've gathered, its acceptability to them, and what next steps would be. And I wouldn't like to second guess that. It's their job to do that. And then we will be present. I'm sure we'll be doing more communication before then, but at the very least, we will be present at the conference that Tanya mentioned at the beginning in Washington in September. And that is what we had to present today. If you have any questions, we're very happy to take them. This is the instructions just here. Otherwise, if you would like to talk to us directly at any time, you can go to www.amo-pharma.com and register to receive automatic updates of any information we release. So that is where we are. Let me see if I can just get myself back on the screen. If anyone has any questions, we'd be happy to take them. Otherwise, thank you so much for listening.
Mike, I think the first question just says, are there any plans to test the drug on adults? And I think you already answered that affirmatively, that is definitely in our plans right now, and that we've put together a provisional protocol for that. We're vetting that at a variety of different levels right now.
Yeah.
I don't know if there's anything you want to add to that.
No, just, it's been an interesting experience. What's that old cliche? If you build a house, by the end of it, you'll have learned how to build a house. I think we're now in a position to understand the lessons we've learned running this study, and we will move into an autism study, an adult onset study, as fast as we can.
And then I was going to say, the next question is, there was an incidence of 7% of falls in the treatment group with AMO02 ? That's correct. So it would be, you all know that postural instability and issues or challenges with walking and ambulation are a core part of myotonic dystrophy, especially early onset myotonic dystrophy. And falls occur fairly frequently. So, but it is true that 2 individuals had instances where they fell. They didn't sustain injury, but nonetheless, it met the criteria that we had defined in our protocol for being an adverse event. And so it was coded as such in that. And it was 2 individuals that were on active drug. And it would have been self-limited, so it wouldn't have been an issue where they had continuing repeated instances of falling several times during the study. These would have been isolated episodes. And then the next question, sorry, Mike, I'm having trouble just scrolling up here.
I can see the next question. It says, do you have a specific time frame for starting a trial? I assume that means in adult onset. No, we don't. The rate limiting step, I would guess, is agreeing the design of that study with the regulators. We are hoping to meet FDA and MHRA around in the fall. Again, that's up to them once we submit the request for a meeting. Dependent on their feedback, we could start a study early next year, but again, we can't go ahead without their blessing. So the timeline will depend on that.
The next question, Mike, says, will Canada be considered for the DM1 study? I presume this is the adult DM1 study. I will answer that myself, which is, yeah, absolutely. Canada has been fantastic country to work with in this REACH CDM program the study that we've just finished. The regulatory agency in Canada, which is called Health Canada, is very accessible and responsive. And the fact that we correspond in English is also quite helpful. Although we did, in our REACH CDM study, we did have a site that was located in Ottawa that, as the Canadian families will know, we're both French-speaking and English-speaking. But, we have very much so enjoyed working with Canada, and I'd love to work with them in the future.
And some of our longest relationships with investigators are investigators in Canada, so I completely agree. Then it says sorry, Jim.
Well, you can read that one. I'll read the next one.
So it says, my son has been diagnosed with congenital DM1 and we live in United Arab Emirates, Dubai. Is there any plan to extend your study into the Middle East area? I'll be honest, not as yet. But if you would like to connect with us over our website or MDF can pass on our contacts, maybe we can discuss your son more one-to-one. Yeah, please do that. That would be the easiest thing to do.
And then the next question, Mike, says, how will what we learn from this AMO2 or from AMO2 help us with adult DM1 and DM2 treatment development? Sorry, there's another part to this I'm trying to read. Will this study help that at all? Would you like me to answer that, or would you like to answer that one, Mike?
I'll go first and then hand that to you. I think we've learned a lot on the execution of the first study that can be generalized. So in terms of clinical trial operationalization, I think any future study AMO Pharma does in adult DM1 will benefit from a lot of learning. There are some very critical things, and in the fullness of time, we will put that out into the public domain. Key lessons learned from how to do studies in this community more broadly, and then theoretically, congenital onset DM1 has the largest repeats, and generally, it's a rule of thumb or a generalization that the more severe the condition, the tougher it is to treat. So if we are blessed with any success in this study and that the efficacy analysis shows benefit, I would have thought, understanding the way the drug works and that we tried it in the toughest situation first, then it should help adults with DM1. DM2 is a separate issue, which I've mentioned and we'll come back to on another occasion. But Joe, please.
Well, I mean, one of the things that I hope would be a learning from the approach we've taken in this study and with this program is that myotonic dystrophy, regardless of the age of onset for the individual, if it's at birth or childhood or in adulthood, it's head to toe symptomatology, head to toe signs and symptoms can be evident in anyone that's affected with myotonic dystrophy. And we have taken an approach in this program where we've gone ahead and tried to assess the full impact of the signs and symptoms from head to toe in affected individuals rather than focusing explicitly on hand grip strength as an example. So I hope that that's a piece that will be influential in a beneficial way to other programs moving forward, that they'll also be thinking about the full spectrum of signs and symptoms that can affect people. And we'll be talking more about that, I think, as our results become available. And yeah, so that's just a piece that I think is also positive and of benefit moving forward from this program. And then
I've just realized we didn't answer one of the questions before. Flora Kim asked, do we anticipate the full results will be presented at the MDF conference? I would hope, Flora, that we present the full results before the MDF conference. It's a relatively short slot at the conference where we could discuss highlights at that time. But in terms of a longer presentation, we will target our aspirations to get it out before that.
The next question, Mike, says, how can my 39-year-old who has MD1, type 1 myotonic dystrophy that appeared at age 25 join the next trial into adult onset myotonic dystrophy?
I think that's Alan, and it looks like, Alan, you're in Melbourne. What we did for the present study is had what we call a study-specific website called the reachcdm.com website. We will do something similar. And the way that worked is it listed all the countries that were involved and some of the criteria required for entry. Every study has inclusion and exclusion criteria. Not everyone can join every study. But as soon as that study is planned and comes into practice, we'll put up a website and that will have facilities to assist people get to a site and so on. So I think, Alan, again, just check back with us as we move forward.
And this next question, Mike, says, if everything goes fine with the study, when is the plan to commercial mass production of the drug?
Again, that's a little bit beyond our control. We will meet with the regulators to present the data that we have, discuss with them its acceptability, whether they think it's enough, whether they think we need more studies, and so on and so forth. So it's a collective effort between us and the regulators. We aim to meet them as soon as we can. We will be submitting data to them and requesting meetings, and we'll take it from there.
And The next question, Mike, says, do you anticipate possibly getting AMO-02 approved to treat even younger patients, less than 6 years of age? I can answer that one, which is part of the agreement that we would reach with the regulatory agencies, whether it's in the United States or in the UK or Canada, Australia, is to account for this younger age segment and to agree to do at least one study in individuals that would be younger than 6 years of age. The design of that study and then the timing of when it would be done would be a point of discussion with the regulatory agencies, but we certainly are interested in making sure that youngsters less than 6 years of age are not that they're included in this program and that they'd be in scope for a drug approval when or if that happens. Let's see, I'm just trying to scroll down to the next one.
Flora's asking about time for release of the analysis, and I think we covered that. And then Maria asks about changing the trial criteria so there's a little more laxity or freedom for people to join the present study. Do you want to speak to what's happening with the REACH CDMX 004 study?
Yeah, we did two things. This extension study that I talked about. So it has a 52-week open label treatment period, which means everybody is on AMO-02. So there's no placebo in that. But we did change the inclusion criteria for that study to include not just congenital onset, but childhood onset, timing of when symptoms started, and as I'd mentioned, the duration of that study has also been extended from 52 weeks to 84, and it's actually going to go longer. But we have some flexibility in terms of who we would bring into that study. The latest amendment that we did to the study, though, was to include childhood onset individuals as well. We broadened the age range also. We didn't go below 6 yet. But as I've mentioned in my previous answer, that's part of our broader plan.
Okay. The next question is from Belen, talking about the relationship between when you take the drug on an empty stomach. Yeah, we appreciate that's a challenge. Often it's the case with medicines, prescription medicines or experimental medicines, you need to be cognizant or careful around taking the drug with or without food. Some of the regulations are quite strict on that. We've got plans to initiate a conversation about whether that can be made easier. I don't think we can talk to a specific target on doing that just in the short term. So that would be a longer term plan. But Joe, please correct me if you think I've answered that badly.
Yeah, I mean, we're not happy. I don't think anybody's happy with the current method that we're using right now about having to have an empty stomach and then taking the medicine and then waiting for a period before you can have your next meal. And that's definitely a point of discussion with the FDA and then the other regulatory agencies. One of the pieces of data that we'll get from this study and the analyses are underway right now is the pharmacokinetic data, which tells us how much of the drug is in the blood of these young people at any given point in time. And then we can compare it against the timing about how long their fast was and when their next meal was to get better insight into whether or not there is a genuine bona fide risk that would unnerve us and unnerve regulators in terms of having too much medicine in their bodies. So we'll be in a position once we're done with that analysis, I think, to have a more informed discussion about methods for changing the protocols to make it more user-friendly for the kids and adolescents and families in that regard.
And then there's a clutch of questions. Two of them, again, they relate to younger children. And I think what I would do if I were you, if you are interested and you have a child younger than 6 years of age, again, is just keep watching for updates from us. Let us get the information out as to whether this trial has conferred benefit. It's important that we establish that in the first instance. If that has happened, then as Joe mentioned, we already have plans in discussion with some of the regulators to push the trial down to younger and younger children in the way that other people have in their drug development. And we will make announcements about that, and then there will be methods. Again, we will always have announcements on our website. You can sign up for automatic delivery of them to your email if you would like. We'll be always presenting at MDF, and we will always have a separate clinical trial website that gives all the instructions, sites where the studies will be taking place, and also individuals on hand. So you can just call them and say, look, I'm interested. I don't know what to do. Someone will, not to put it patronizingly, hold your hand and take you through the process. And then in terms, again, I think I've answered the question. We would love to have another webinar if the MDF can help us. As soon as we have the results to do a conversation like that, then yes.
And then the question about how should we follow you to get updates, I think we'll try to put timely updates on our website, and then we'll also stay in touch with the MDF as well, and so that they'll be apprised of what it is that we're doing and how we're doing in terms of results analyses. And then the next question says, are you hopeful that you're moving closer and closer to a cure for myotonic dystrophy? Do you want to try that, Mike?
That means we always have hope. I think we need to be careful. Logically, and from the evidence we know, there's been encouragement in the previous trial. What we would love to see is further encouragement from this study. And if there is further encouragement from this study, then yeah, we've taken a step closer to a potential treatment. I'm sorry if I'm being a scientist from a drug company. I get nervous about using the word cure, but I think we are taking steps in the direction of a treatment. But let's wait and see what the next data readout is like, which again, we will share with you as soon as we're able to do that.
But I do think, I mean, I would add though to this that the more and more companies and different sponsors have become interested and involved in research in myotonic, both type 1 and type 2 myotonic dystrophy. And it's just beneficial from a scientific viewpoint and again, a positive humanitive effect for affected individuals and their families, because the more resources that are put forth in a particular therapeutic area, particularly a rare or orphan therapeutic area like this, I mean, it's just good that comes from that, because best practices around how to design studies, what to measure, education of the regulatory agencies, agreement on what sort of designs are optimal. All of that is coming from this increased interest that we all collectively have in doing research and doing clinical trials in myotonic dystrophy. And I think we're contributing to that with the AMO 02 program. So in that regard, yeah, I mean, I think if you look back compared to 10 years ago, so even 5 years ago, where we're at right now in 2023, we're in a far more advanced or closer place with regard to even thinking about the word cure than we were, again, 5 to 10 years ago.
I would say it's a virtuous circle as well. Every time anybody does a study, it contributes information which is helpful. Equally, participating in a study, from our perspective, the ability to engage with the community, the opportunity to be involved is incredibly energizing for us. So I think, the worst situation is nothing happening. When lots of things are happening, it feeds on itself in a positive way.
Tanya, do you have additional questions? Because I think I've scrolled through everything that I can see here, but I don't know if there are other questions that have been asked that we haven't answered.
No, we don't have any other questions that have come in to us. So I think if you are feeling good about the presentation and have answered all the questions, then I want to thank you both for the very comprehensive overview. And of course, MDF will be very excited to host another webinar when you have data to share. We look forward to hearing about, in the coming months, what the outcome is, as well as at the conference in September. For those of you who will be at the conference, AMO is one of our sponsors, and they will be there to answer questions as well as to do a very short presentation. I want to thank everyone for being here today. And Dr. Horgan, Dr. Snape, thank you so much. We hope everyone has a fantastic weekend.
Yep, thank you, everybody. Thanks for letting us do this today. We really appreciate it. Have a great weekend.
No, thank you, Tanya. Absolutely. And thank you to everyone who's taken the time to let us speak today. We appreciate it.